Welcome to DNA barcoding molecular biology research at Valencia. We started in May 2017 and it's been going on ever since. Today is June 26, 2022, summer, and my name is Nalini Odapalli. You can call me Professor O. The way this research goes, molecular biology research, quickly explains that we start from the first step, scratch, which is collecting the plant tissue from places that we select with Lake Pamela at the West Campus of Valencia. With a group of students, we go with their gloves and Ziploc bags and Sharpies, and we went to other places. Sometimes we collected samples from our backyard. This is in Park Garden. We collect the samples and we bring it back to lab, wherein we clean the tissue. I give the protocol and I explain what to do. I demonstrate, and then we do something called PCR. After cleaning the leaf tissue, we do polymerase chain reaction, which is making multiple copies of the DNA. This is DNA barcoding, right? So this is molecular research, molecular biology research. After we extract the DNA, there are so many small ingredients. How would we know that we extracted the DNA and it is there and we did not damage it, right? So we do the next technique, step 4, called electrophoresis, which is take the solution and put it in these little slots called wells in the gel. It is called polyacrylamide gel or agarose gel. It allows the DNA molecules to go through its network. Yes, it looks like a block, but it is not solid block. It has fibers and passages, and we run electricity by putting this gel with the DNA in a solution. And electricity, yeah, it doesn't damage the DNA because DNA is negatively charged. Probably you do not know this. What happens when we run electricity by putting DNA in the gel is DNA runs towards positive electrode. But then in this process, we separate the different size DNA fragments. This allows us to confirm whether DNA was isolated properly and then it has been kept intact. Then the next step is we take the DNA and we ship it to company. Yes, DNA can be shipped. It won't be damaged, to a company in New Jersey called GENEWIZ. They do gene sequencing. What is gene sequencing? I want you to understand that a gene molecule is made of small molecules and they can be identified. If I see this cell phone, I can say that this is black colored cell phone. If I see the paint, I can say it is green, my shirt is red. Like that, molecules can be identified by the machine and they are read in the form of letters in order. And as you can see here, this is called DNA sequencing or DNA sequence, which they do at their end. They have that company, gene sequencing machines, and they send that data by email because now it is data, right? And we take the data and we put it in bioinformatics tools. What is bioinformatics? Combination of computer applications and biological data. We use the software to analyze the data and make sense of it, understand what's going on, many things about it. This bioinformatics tool is called the Basic Local Alignment Tool, or BLAST. And this is a software program which will help us understand the relativity of this particular species sequence with many other species. And by using another tool available online for free for students called DNA Subway, yeah, it's like Subway sandwich, but it is called DNA Subway, we generate phylogenetic trees. That means our sample, what is it closely related to, so that we can understand its genealogy. Phylogeny is called. So this is our 2018 team. We presented all our research. Each individual student has done their research with individual species and came up with the trees and analysis, so much more, which I'm not covering here, at a research conference called PLUCK, Florida Undergraduate Research Conference, which was conducted in Melbourne. And we drove down there. We stayed for a day, all funded by Valencia College. And LSAMP especially. You can see all these are the individual posters of Jasmine and Alejandra, and Brian, Balaji, and one of my students, she earned a first prize at from Lake Mary College here, Seminole State College, and she even went to Arizona taking this research to annual biomedical research conference. So this is a poster. This is what we come up with, all the data, and we put it together. We perform or present this data or a posters printed really for Valencia students at the undergraduate research conferences at UTF, Valencia, and so many places. You can read more from this slide, pause and read the slide. This is one credit hour course, and you need to contact me at nodapalli@valenciacollege.edu. You can also call me at 754-255-2082. If it is in person, then we conduct it on Fridays. Keep your Fridays free if you are planning to join my research. Molecular biology research, it reflects so highly on your resume. I will stop with this information. There is more and more information how to get ready for future. How does it help? If you get a stipend, there are hospital internship opportunities, lab assistant opportunities at Valencia. International, national awards can be won. People have won awards using this kind of college research. You can meet me. I can guide you on how to obtain a research opportunity or further go with this experience, and how to get other internship opportunities at UCF. I'm a graduate of UCF myself. How this will help you to showcase and show off when you're applying for jobs. I will train you along the way with all of this.